Hi everybody. Today we shall discuss about some of the questions from the chapter Cell Cycle and Cell Division. Cell Cycle and Cell Division is one of the important chapters from which usually two or three questions are asked in NEET. So let us discuss some of the important questions that can be asked from this chapter. Before we go into the details of the questions, please subscribe my channel, share it, comment it, and also hit the bell icon to get the notifications as soon as I publish the various types of question papers with solutions in this channel. So let us enter into the questions related to this particular chapter. The first question is, pea plant has 14 chromosomes in each cell. The 2n means diploid one. If the number of linear DNA molecules in G1 phase is 14, what would be the number of DNA molecules in G2 phase of the cell? So it has got 14 chromosomes. In G1 phase, every chromosome will be having only one chromatid. One chromatid means one DNA molecule. So in G1 phase, the diploid pea plant cell has 14 DNA molecules. In S stage, there is synthesis of DNA. At the end of S stage, the amount of DNA is doubled. Hence, the 14 DNA molecules will become 28 DNA molecule. Each DNA molecule is present in one chromatid. A chromatid has a chromatid, the chromosome has the two chromatids at the end of S stage. So whatever the chromosome number that is seen in G1 phase is continued even in S phase and G2 phase. But the number of chromatids and DNA molecules are doubled at the end of S stage and it is continued even in G2 stage. So now the total number of DNA molecules that are seen in the diploid cell will be 28. Let us see the answer. Answer is 2. Let us see the explanation. In G1 phase, each chromosome possesses only one chromatid and each chromatid has only one DNA molecule. The number of linear DNA molecules would be 14 in this stage. In the next stage, that is S stage, the amount of DNA is doubled due to replication of DNA. At the end of S stage, each chromosome biochemically possesses two chromatids and each chromosome with two DNA molecules. Hence, the number of chromatids and DNA molecules become 2 into 14 or double the number of 14, that is 28. The same number of DNA molecules is continued in G2. Hence, the number of DNA molecules in this stage remains 28 as there are 28 chromatids. Coming to the next question. Arrange the following events of cell cycle of animals in correct sequence. Separation of sister chromatids of chromosome. So this separation is done in anaphase. Duplication of most of the cell organelles, that is in G1 stage. Duplication of nucleus. It happens in telophase. Daughter nuclei are formed in telophase. Duplication of centrioles. This takes place in S stage of interphase. So, among these four, first one, duplication of most of the cell organelles, which is in G1 stage, B first. Next, the duplication of centrioles in S stage, B, D. Next one is separation of sister chromatids of chromosomes, that is A. So, the last one is duplication of nucleus in telophase, that is C. So, the answer must be B. The duplication of most of the cell organelles. D. Duplication of centrioles. A. Separation of sister chromatid chromosomes. And C. Duplication of nucleus. The answer must be the second option. Let us see. Given. Here it is explanation. Very clear in the explanation. Whatever I discussed, everything is given properly in the explanation. Right? The cell cycle has various stages in a sequence, namely G1, S, G2, and M stage that you know. The G1 stage, most of the cell organelles are doubled. In S stage, the centrioles of cell are doubled. In anaphase of M stage, the sister chromatids of each chromosome are separated. In telophase of M stage, two daughter nuclei are organized. Coming to the next question. Statements. Statement 1, Statement 2. Duration of cell cycle differs from cell type to cell type of an organism. Absolutely true. A cell usually withdraws from cell cycle at given stage. This is also true. Both the statements are correct. Right? So, what option must be the answer? Both statement 1 and statement 2 are correct. The fourth option must be the correct one. Let us see the answer. Fourth one. Explanation for this. So the duration of cell cycle varies from cell to cell of an organism. Duration of cell cycle of shoot apical meristem, an example is given here, is lesser than that of the root apical meristem. Yes. A cell usually withdraws from cell cycle at G1 stage and enters into quiescent stage or G0 stage. Coming to the next question. Which permanent cells of growing woody dicot can enter into cell cycle from G0 stage? So it is something related to the entry of permanent living cells into cell cycle. Permanent living cells enter into cell cycle only during dedifferentiation. So then we have to identify which tissues re-enter into cell cycle during dedifferentiation in a woody dicot at the time of secondary growth. So first one, cortical cells of stem, absolutely true. Cells of the cortex of the stem dedifferentiate and form corticambium. Right? So during this formation of corticambium, cortical cells enter into cell cycle from G0 to G1 stage. Medullary ray cells, absolutely true. Those medullary ray cells present between fascicular cambia of adjacent vascular bundles re-enter into cell cycle to form interfascicular cambium. This is also true. Cells have conjunctive tissue. Yes. Cells have conjunctive tissue present beneath the phloem of dicot root. Enter into cell cycle to form bulk of the vascular cambium of root. Cells of root pericycle. This is also true. Cells of the pericycle present opposite to protoxylem of root enter into cell cycle to form the remaining little vascular cambium other than that is formed by the conjunctive tissue. Complete pericycle is also involved in the formation of corticambium. Hence A, B, C, D are correct. Option 4 must be the correct one. Let us see the answer. It is 4. Look at the solution. Cells of medullary rays present between fascicular cambia of adjacent vascular bundles re-enter into cell cycle to form interfascicular cambium. The cortical cells of dicot stem, usually parenchyma, re-enter into cell cycle to form corticambium. In dicot root, the cells have conjunctive tissue present below the phloem enter into cell cycle to form bulk of vascular cambium. Cells of root pericycle re-enter cell cycle to form a part of vascular cambium and a total corticambium. Coming to the next question. Assertion reason type. In a flowering plant, both haploid and diploid cells divide by mitosis. Absolutely true. Both sporophytic cells and gametophytic cells divide in flowering plant. During the formation of gametophyte from respective spores, microspores during formation of male gametophyte, megaspores during the formation of female gametophyte or embryo sac. They divide. And the sporophytic cells also they divide by mitosis. In a flowering plant, meristem helps in the growth of the plant. Absolutely true. That is growth of the meristem helps in the growth of the plant by showing mitotic divisions. And generative cell of the male gametophyte at two-celled stage forms two male gametes. So this is also true. So both assertion and reason are correct. And the reason clearly explaining that the diploid cells and haploid cells, both of them of the flowering plant divide by mitosis. So A is correct, R is correct, and R is the correct explanation of A. Fourth option must be the correct one. Let us see. Fourth option is the correct one. Here is the explanation. Let us enter into the next question. It is a matching type. Carefully look at this one. Movement of chromosomes towards equator of the cell. Toward the equator of the cell, the chromosomes move during early metaphase or metaphasic stage. So A3. There A3 is the first option, third option. So you can omit second and fourth options. Chromatin becomes chromosomes. Chromatin is organized of chromosomes in prophase. 4. A3, B4. So it looks like the first option must be the correct one. Chromosomes become chromatin network, that is in telophase. 1, C1. Chromosomes movement towards poles of cell. A chromosomal movement towards poles of the cell, where in anaphase. 2. So the first option must be the correct one, that is A3, B4, C1, D2. Let us see the answer. Answer 1. Here is the explanation. You can have a look at it. Coming to the next question. Identify the correct statements with respect to mitosis. With respect to mitosis, you have to identify the correct statements. Spindle fibers are attached to kinetochores of chromosomes during prophase. Wrong. It is wrong statement. Spindle fibers are attached to kinetochores of chromosomes during metaphase, not in prophase. So A is wrong. So you can eliminate first one and second one. So third and fourth one, out of these two, one must be the correct one. Daughter centrosomes move towards opposite poles during prophase. This is also true. The daughter centrosomes move towards opposite. Centrosome divides into daughter centrosomes in S stage of interphase. Those daughter centrosomes, with each daughter centrosome having two centrioles, move towards opposite poles during prophase. This is true. Chromosomes are aligned at the equator in metaphase. This is also true. Daughter nuclei are formed in telophase. Absolutely true. Sister chromatids are referred as daughter chromosomes in anaphase. Yes, they are referred as daughter chromosomes in anaphase upon separation from each other due to splitting of centromere. So B, C, D, E are correct. The fourth option must be the correct one. Let us see the answer. Fourth one. Here is the explanation and also read out. Spindle fibers are attached to kinetochores of chromosomes during early metaphase. Daughter centrosomes with a parental centriole and a daughter centriole move towards opposite poles in prophase. Chromosomes are brought to equator by chromosomal spindle fibers in metaphase. Daughter nuclei are formed in telophase with the reorganization of nuclear envelope around chromosomes of respective poles. The two separated sister chromatids of anaphase are referred as daughter chromosomes. Coming to the next question. You have to choose the correct combination here. Cell plate, cytokinesis of animal cells. No. Cell plate is not formed in cytokinesis of animal cells. Cell plate is formed during cytokinesis of plant cells. So it's not correct combination. Syncytium, a cell with several chromosomes. No. Syncytium is a cell with several nuclei. It is also incorrect. Metaphasic plate, plane of alignment of chromosomes. So it is a plane of alignment of chromosomes during metaphase is known as metaphasic plate. Right? So third option must be the correct one. Chromosome condensation. Chromosomal condensation is seen in prophase, right? Not in anaphase. No further condensation of chromosomes in anaphase. Whatever condensation that has to be done, that will be done in prophase or till the earliest stage of metaphase or transition stage between prophase and metaphase. So option 3 must be the correct one. Let us see the answer 3. Here is explanation. Cell plate, it is formed by Golgi vesicles during cytokinesis of plant cells, not animal cells. Syncytium, a cell with several nuclei formed either due to fusion of cells or division of nuclei without cytokinesis. Chromosomal condensation, prophase. Metaphasic plate, plane of alignment of chromosomes in metaphase, which is also known as equatorial plate. Next question. Choose the one not related to mitosis. Which is not related to mitosis, you have to choose. Pairing of homologous chromosomes, not at all related to mitosis, it is related to zygotine of prophase 1 of meiosis 1. So 1 must be the correct answer. Attachment of chromosomal fibers to kinetochores. It is related to mitosis as well as meiosis. A checkpoint for ensuring attachment of all chromosomes with spindle fibers at the centromere. Yes, that checkpoint is present between metaphase and anaphase. Transition to anaphase from metaphase. That is there in mitosis. Deformation of Golgi complex in telophase. So it is related to mitosis. Reorganization of Golgi complex endoplasmic reticulum is seen in telophase. So first one must be the correct answer. Let us see the answer. 1. Here is the explanation. Pairing of homologous chromosomes is observed in zygotine of prophase 1 of meiosis 1, but not mitosis. Chromosomal fibers are attached to kinetochores in early metaphase. Before the entry of cell from metaphase to anaphase, the attachment of spindle fibers to kinetochores of all chromosomes is ensured by a check which is known as spindle assembly checkpoint. Golgi complex is vanished in prophase and reformed in telophase. Next one. Nucleocytoplasmic ratio of a cell is disturbed during interphase due to cell growth. Increase in the protoplasmic content, increase in the size of the cell. Right? Nucleocytoplasmic ratio of a cell is disturbed, right, during interphase, not during cytokinesis. Cytokinesis is restored, not during metaphase, not during prophase, it is interphase. Let's see the answer, must be the fourth one. Fourth one is the answer. So due to active metabolism or RNA and protein synthesis or DNA replication, interphase, the nucleocytoplasmic ratio, so the ratio between the volume of nucleoplasm to cytoplasm is disturbed and volume to surface area is also disturbed, right? So there is more increase in the volume than the surface. This is also disturbed. So whatever that is disturbed due to cell growth is restored as a result of cell division. To restore it, the cell divides and at the end of cytokinesis, it is totally restored. Coming to the next one. Again, statements 1 and 2 are given. You have to identify which is the correct one, which is the incorrect one, or both are correct ones, or both are incorrect ones. Plasmata seen in diplotin, plasmata seen diplotin and meiosis indicate the sites of crossing over. What are plasmata that are seen? Diplotin or meiosis indicate the sites of crossing over. Absolutely true. Crossing over takes place in diplotin, wrong, takes place in packaging. Statement 1 is correct, statement 2 is wrong. Statement 1 is correct, but statement 2 is incorrect. The first option must be the correct one. Let us see. Answer 1. Here is the solution. Chiasmata are clearly seen in diplotin of prophase 1 due to degeneration of synaptonymal complex and moving away of homologous chromosomes or bivalents without movement at points of crossing over. Chiasmata are physical proofs for crossing over which take place in pachytene due to the action of recombinase at the recombination nodule region. Coming to the next one. The assertion reason time. Let us have a look at it. Paired homologous chromosomes are zygote together referred as bivalent. Absolutely true. Together referred as bivalent. Each chromosome of bivalent has two chromatids. Yes, this is also true. Each chromosome of bivalent has got two chromatids. Right? But the reason is not explaining the assertion. Bivalent is pairing together of the two homologous chromosomes together known as bivalent. Not by just having the two chromatids in each chromosome, we don't call it as bivalent. Right? So assertion is true, reason is also true, but the reason is not explaining the assertion, so answer must be the 2. 2 is the answer. There is a solution. The paired identical maternal and paternal chromosomes in zygote stage of prophase 1 together referred as bivalent. The bivalent which has 4 chromatids, 2 chromatids of each chromosome is also referred as tetrad, which is very clear in packaging stage. The 4 chromatid nature of the bivalent is clearly seen in packaging, though bivalents are formed in diplotin. That is about the solution. Coming to the next one. Which stage of meiosis the centromere splits and daughter chromosomes are formed? So anaphase 1, it is a separation of maternal and paternal chromosomes, which is known as segregation or disjunction. Anaphase 2, bivalents or chromosomes are aligned at the equatorial region. Anaphase 2. Anaphase 2, yes, in anaphase 2, the centromere splits and the two sister chromatids of each chromosome are separated. Hence, daughter chromosomes are formed in metaphase 2. So, it must be the anaphase 2. It must be the correct answer. Anaphase 2 must be the correct answer because centromere splits and daughter chromosomes are formed in anaphase 2. The third option must be the correct one. Metaphase 1. No, metaphase 1 bivalents are aligned at the equator. So, the answer must be third option. Let us see. Third option is the correct one. Let us look at the solution. Anaphase 2, the chromosome splits at the centromere and daughter chromosomes are formed. In anaphase 1, the maternal and paternal chromosomes of each bivalent are separated. In metaphase 2, all the chromosomes are brought to equatorial region by spindle fibers. In metaphase 1, the bivalents are brought to equatorial region of cell. Coming to the next one. Matching. Match 1, list 2 must be matched. Each chromosome with only one kinetochore. Each chromosome with only one kinetochore is seen in anaphase 2. A4. A must be 4. A4, 2, 3, 4. First is eliminated now. Option. Each chromosome with two kinetochores. Each chromosome with two kinetochores. That each chromosome with two kinetochores is in anaphase 2. Right? So, A. Anaphase, each chromosome with two kinetochores. Each chromosome with two kinetochores is in anaphase 1. So A, each chromosome with only one kinetochore, anaphase 2, 4, A4. B, each chromosome with two kinetochores is anaphase 1, that is 3, A4, B3. After that, centriole duplication. Centrioles duplicate in interphase or interkinases. Right? Interkinases centriole duplicate. C2 and haploid nuclei with a 2C amount of DNA are formed in telophase 1. Whereas in telophase 2, 1C DNA. Haploid nuclei with 1C DNA in telophase 2, 2C DNA in telophase 1. So, what must be the answer? A 4, B 3, C 2, D 1. Fourth option must be the correct one. Let us look at the solution. Chromosome with only one kinetochore, anaphase 2, due to division of centromere. Chromosome with two kinetochores, anaphase 1. Centriole duplication, intrakinesis between meiosis 1 meiosis 2. There is no DNA replication, but centriole has to be duplicated in animal cells. That information is not given in the book, but has to, otherwise, while polar spindle is not formed. Haploid nuclei with 2C DNA formation, telophase 1. Coming to the next one. A is the stage of meiosis during which the separation of paired homologous chromosomes is initiated. Separation of homologous chromosomes are bivalent is initiated in diploidy due to degeneration of synaptonymal complex and establishment of repulsive forces between the two chromosomes are bivalent. And B is the stage at which the separation of homologous chromosomes is completed. Separation of homologous chromosomes is completed in anaphase 1. A and B are respectively must be diploidy and anaphase 1. Fourth option must be the correct one. Let us see. Fourth one. So, explanation very clear in the solution. With the degeneration of synaptonymal complex and establishment of repulsive forces between homologous chromosomes in diploidy of prophase 1, the separation of homologous chromosomes is started and they are completely separated towards respective poles in phase 1 of meiosis. Coming to the next one. Arrange the following events of meiosis 1 in correct sequence. In perfect sequence, we have to identify or arrange. Perfect sequence. Meiosis 1 events, all these are, you have to remember that. Terminalization in early diakinesis. First event in diakinesis, which is the last substage of prophase 1, is terminalization. Bivalent formation, zygotine. Genetic recombination, packetine. Vanishing of nuclear envelope, last step in diakinesis. Bivalence at the equator in metaphase. Or metaphase 1, you can say. So, first event is bivalent formation, B. You have to start with B, bivalent formation. Next is C, genetic recombination in packetine, right, due to crossing over. Right, B, C. After that, terminalization, BCA, terminalization. After that, vanishing of nuclear envelope, D. After that, bivalence at equator. So, first option, B, C, A, D, E must be correct. B, C, A, D, E. First option must be correct, first option. Come to the explanation. Bivalent formation or synapses, zygotine. Genetic recombination, packetine, recombinase activity. Terminalization, first event of diakinesis, movement of chiasmator towards telomeres. Vanishing of nuclear envelope, last event of diakinesis. Bivalence at equator, metaphase 1. Coming to the next one. Which stage of meiosis can last for years together in oocytes or some vertebrates? So, one of the stages of prophase 1 can last for days, months and years together in oocytes of some vertebrates, even human females. Right? Diakinesis, no. Meiosis 2, no. Packetine, no. Diploidy, only in diploidy. In diploidy, in oocytes of some vertebrates, there is also at certain major regions or of the chromosomes or diploidy, there is decondensation or despiralization or decompaction. As a result, at that particular regions which are usually euchromatic regions, there are extensive synthesis of RNA, which also leads to the formation of proteins later. Extensive synthesis of RNA is seen. High rates of transcription. Chromosomes of that stage in diploidine, in amphibian oocytes or the vertebrate oocytes, the mammalian oocytes also, appear like a lamp brush. Hence, chromosomes, such chromosomes very larger in size and referred as lamp brush chromosomes. Right? Let us see the answer. It's multiple diploidine. It is also given in the book. Answer 4. Diploidine or meiosis can last for years together in oocytes of some vertebrates. During diploidine, the chromosomes show decondensation at certain regions, and at these regions, extensive RNA synthesis or transcription is observed. Chromosomes of this stage are larger and appear like a lamp brush, and such chromosomes are called lamp brush chromosomes, which are one of the giant chromosomes. Coming to the next one. Which stage of meiosis represents transition to metaphase 1? Which stage of meiosis represents transition to metaphase? It must be one of the stages of prophase 1. After prophase 1 only metaphase 1. The last one in diakinesis. So, last one in prophase 1 is diakinesis. It must be the answer. Packaging, no, it is somewhere in the middle. Diakinesis, yes, it's the last one. Second is option is the correct one. Diploidine, no. Zygotine, no. Diakinesis, answer 2. Diakinesis represents a transition to metaphase 1 of meiosis with the degeneration of nuclear envelope. Coming to the next one. Choose the incorrect statement. How to choose the incorrect statement. Meiosis helps in conserving the same chromosome number of a species across generations in sexually reproducing organisms. Thus, this is the correct statement. Meiosis helps in maintaining constant chromosome number in every generation of sexually reproducing organisms. If there is no meiosis in sexually reproducing organisms, in every generation the chromosome number is double. Because meiosis is not there, there is a formation of only diploid gametes. When diploid male and female gametes fuse, tetraploid zygote will be formed. Ultimately, tetraploid individual is developed. Again, next generation to become octaploid. After that, 16N, 32N, 64N, like that. Right? 128N, like that. So, but meiosis helps in reducing the chromosome number to half. So that it helps in the formation of haploid gametes. Directly or indirectly. The correct statement. Meiosis brings about genetic variability due to random distribution of maternal and paternal chromosomes of bivalence to daughter cells. Yes. During meiosis particularly, the alignment or distribution of maternal and paternal chromosomes of each bivalent independent of another one. In one bivalent, a maternal chromosome towards upper pole. In another bivalent, it may be towards the lower pole. In the third one again it may be upper pole, fourth one again it will be lower pole, fourth bivalent. So this also brings about genetic variation because random distribution of maternal and paternal chromosomes brings about genetic variability. So this is also correct statement. The event of packaging helps in bringing about genetic variability in the offspring. Yes. The exchange of genetic material or DNA between maternal and paternal chromosomes are bivalent. It also brings about genetic variability. Third one is also correct statement. Naturally, the fourth one must be the incorrect one. Each daughter cell formed after meiosis 2 is haploid, true, up to here it is true, and has 2C amount of DNA, now 1C amount of DNA. The haploid nucleus that is formed after meiosis 1 has got 2C amount of DNA. After meiosis 2, each daughter nucleus will be having 1C amount of DNA but not 2C. The daughter nuclei of the meiosis 1 haploid, 2C amount of DNA. The daughter nuclei of our daughter cells are formed after meiosis II will be having haploid number of chromosomes and haploid amount and 1C amount of DNA but not 2C amount of DNA. So fourth option must be the correct one. Coming to the answer, fourth one must be the correct one. Coming to the solution. Each daughter cell formed after meiosis II is haploid and has only 1C amount of DNA, whereas meiosis I daughter nuclear daughter cells will be having 2C amount of DNA, though haploid. What is the common between these two? Haploid, haploid. What is the difference? Meiosis I daughter nuclei will be having 2C amount of DNA. Meiosis II daughter nuclei will be having 1C amount of DNA. That means the amount of DNA is cleaved to half or reduced to half after meiosis I as well as meiosis II. Coming to the next one. What is the shape of chiasmatas in diploteinomeiosis? Chiasmata are cross-shaped structure. So cross shape is X shape. Or Y shape, M shape, V shape, X1. Fourth option must be the correct one. Let us see the answer. Fourth one is correct. Chiasmata appear as X-shaped structures which are clearly seen in diplotein due to disappearance of synaptonemal complex and also slight moving away of both the chromosomes without the movement of chiasmata. So that's all about the questions from this chapter. All are typical questions. Carefully you follow, repeatedly you see and recollect.